Good afternoon. So we are going to simply discuss about ROS in human physiology. Human physiology means our biological, our human body may, what are we going to, where are we going to see reactive oxygen species, we are going to go through. So all of us would have been heard of lipid peroxidation. Lipid peroxidation is the most common term we all would have heard. So in the same way, the most common reactive oxygen species that we are going to see in body fluid is prostaglandins. So that has more applications from medical point of view. So prostaglandins are actually synthesized from the lipid, phospholipids are present in the cell membrane, right? So from there they will be synthesized. So from the phospholipids, arachidonic acid will be formed, and then this will be the precursor that will be acted upon by two major enzymes. Cyclooxygenase forms two peroxides, and further action of peroxidase will lead on to the major precursor PGH2. So there are some 78 types, grossly 78 types of prostaglandins. So PGH2 will be the precursor. So these prostaglandins are going to be associated with inflammation or there are some minute triggers that will be causing the release of prostaglandins. For example, there is some allergy, dust. So those times there will be bronchial constriction and cough. So during that times prostaglandins will be secreted in bronchus. So whenever you get hungry, there will be stimulation by histamine. So there will be mucus secretion in the gastric region. So that will be due to prostaglandins. So any small insult, prostaglandins will be released. So there are, I told right, there are some 78 types of prostaglandins. So of these, PGE2, PGF and PGD are major. PGE2 and PGF2 alpha are major. PGE2 will be involved in many places. Even during pregnancy, PGE2 and PGF2 alpha are used as medications. They will be giving medications to facilitate smooth muscle dilatation and contraction respectively. At the outlet, there will be dilatation, inside uterine contractions. For these things and all, they are most commonly using these medications. The main purpose will be to increase vascular flow. For example, due to any insult or irritation, there will be increase in vascular permeability. So there will be bleeding or leaky vascular channels. So during those times, prostaglandins will be acting to clot. So at most places, it will be increasing the clot formation, except PGI. So PGI and TXA are one combination. So TXA is main platelet activator and clot formation. PGI is opposite of that. So all the prostaglandins will be increasing the clot formation except PGI. The paracetamol tablet that we are using, right, it is going to inhibit this cyclooxygenase. Cyclooxygenase will be inhibited by paracetamol so that this clot formation doesn't occur. Whenever there is a clot, we have to calibrate it. If the clot is too much, there will be swelling, things like that, right? So it will be relieved when we give paracetamol. So prostaglandin is natural mechanism where clot is formed and there will be some swelling. So natural healing is occurring. So to calibrate it, we give paracetamol. So it will slightly dilate the vessel so that the blood flow continues. So pain also releases. We can continue our work during minor infections. So the research papers that actually mentioned is the next step, the next level, which showed that how prostaglandins associated with tumor progression. Generally, increased free radicals, we know that it will also be causing oxidation of many transcription factors, proteins, so that generally tumor progression is accelerated and at the end stage the cell will die. So if you increase the ROS levels, the cell may go for death or may progress for tumor. So these two prostaglandins are generally don't have any physiological function. Normal physiological functions won't be there for this PGJ2 and PGA2, but they are involved in inhibiting some normal antioxidative mechanisms in the cell. For example, this thioredoxin reductase mechanism will be impaired due to this free radical action. They will be forming covalent adduction, covalent modification of these proteins and enzymes, so that this normal function is impaired. So transcription factor like P53 that suppress tumor will not function properly. They all will function only in the reduced state. But this is not the only way. There are also other ways where these prostaglandins also, in the same way as free radicals, they will also inhibit some other proteins and peptidases, so that the cell might go for apoptosis. So in this way it will be useful for cancer therapy. So in that way there is another paper which showed that they inhibit some peptidases which are going to degrade ubiquitin polypeptide combination rate, so which will be responsible for removing damaged proteins. Removing damaged proteins is one way very important. So this ubiquitin polypeptide combination will be degraded by this peptidase. So this peptidase will be inhibited by some prostaglandins. So there are some two major researches going on. So one is acceleration and one is for cancer therapy. So following prostaglandins, we will be seeing two other most common free radicals in human physiology. Other two things actually we all would know, because one is hydrogen peroxide, because peroxidase and peroxides, this system will be responsible mainly for combating microbial infections. So microbial infections for combating, you will be forming hypochlorous acid inside the neutrophils. They call it oxidative burst rate, oxidative burst reactions during infections. So during these times, hypochlorous acid will be formed from H2O2. The initial steps will be formation of hydrogen peroxide from superoxide radicals. As we know, from superoxide radicals, H2O2 will be formed by superoxide dismutase, and then from H2O2 we will be forming HOCl. And another thing that you should notice, for synthesis of normal thyroid hormone requires iodine. We all know that iodized salts are becoming popular just because of this thyroid deficiency. So this will be combated, and this synthesis involves formation of iodium ion, I plus. Okay, I plus will be formed from I plus will be formed from I minus. So I plus will be formed, and this I plus will be together joined with thyroglobulin. From thyroglobulin, you will be forming monoiodo and diiodo-tyrosine, and then thyroxine, biologically active thyroxine will be formed. So this step involves the action of thyroid peroxidase to form hypoiodous acid. Hypoiodous acid have plus charge on iodine. So the last major free radical is nitrous oxide. So nitrous oxide is actually a free radical scavenger in biological system more than a free radical. So it quenches the free radicals present in the biological system. So it will be synthesized from proteins, arginine, NADPH, and molecular oxygen. So these things will be involved in synthesis of nitrous oxide. So nitrous oxide will be involved in blood flow, increased blood flow, same as other free radicals, and it will also prevent platelet aggregation, in contrast to platelet adhesion by prostaglandins. So here it will be causing it will be preventing platelet aggregation, and it is actually protective during atherosclerosis, endothelial injury. So these instances it is protective. This is one major important area where reactive oxygen species play a role, because you all see disulfide bonds, right? Disulfide bonds are being formed in between the peptides. cross-links are getting formed. So many cross-links are formed between many larger proteins. So for these instances, oxidative cross-linking is important. For example, even collagens, it is very important. And whenever there is abnormalities due to excess reactive oxygen species, abnormal collagens will be synthesized. Abnormal collagens will be resulting in aberrated repair. So that will be seen in diabetic foods. And those instances, the collagen formed will be very different, so that it cannot be degraded and it will not be uniform, so that the healing will be ununiform, so that the ulcer will not heal. So this is major importance, as well as there is some turnover happening in our body. So that turnover will be disturbed, so that there will be more collagen formed and it will be deposited in many tissues. 1 minute. There will be more collagen will be deposited in many tissues. So such things will be causing like glomerular sclerosis, diabetic nephropathy, and then lens cataract. These instances are all mediated due to excess oxygen mediated pathologies. So these things I took from the Barry Halliwell only. You can go through references. There are some reference papers there in the references. So it is present in the book. So I just went through and then I took these. So for this, we can use antioxidants, and these are being prescribed in hospitals. Some antioxidants are particularly N-acetylcysteine will replenish glutathione. Okay, that's it. That's it. Yes.
So is it a stable molecule or unstable? Because as far as I know, the byproduct of lipid peroxidation will again be a free radical which mimics the pathway again. Is it a stable or unstable?
The final end product is stable.
Stable.
Because I told right, the first step will be cyclooxygenase forming the peroxides. There will be one cyclic oxygen, and then one more place there will be peroxide. It will be soon removed by peroxidase. So that you will be soon having the precursor PGH2. So even that cyclooxygenase, that cyclic part is there, right? That will also be degraded in further steps while forming the other prostaglandins. So lipid peroxidation is the initiator. So and then the final products are stable, as long as it is getting eliminated by our body.
But as you said, it's been seen in case of blood clotting. Can it be used as a marker for detecting any thrombosis formation in people in a much earlier stage? Is it possible to check?
How fast the thrombus is getting formed?
No. Is it possible to have this as a marker to detect any people who are prone to thrombosis in their future?
Actually, more than them, there are clotting factors, right? Those clotting factors will be analyzed in vitro. So that will be more important in assaying. You can go through thromboxane A2, right? I told, right, this combination PGI and thromboxane A2. Thromboxane A2 is involved in clotting. So even with that, they won't be analyzing it for how thrombus is getting fastly formed. So that sort of things are not there. They are just triggers. They will be used like, can we inhibit it and reduce the thrombus formation, like that they will be doing. But to test the speed, they won't be using it. Prostacyclin, you call it as PGI. PGI is called as prostacyclin. Prostacyclin is involved in vasodilation. Because there are many other potent vasodilators, actually PGI is not recommended. It is just physiologically present. If you want to know one, aspirin is the major antiplatelet. Aspirin inhibits synthesis of thromboxane A2. I told, right, the cyclooxygenase enzyme inhibitors are more common. Aspirin, paracetamol. So that will be directly given, so that there will be no clotting. Defective collagen. Actually, defective collagen, yes, defective collagen means many cross links will be formed. Yes. Because glutathione is an antioxidant, right? N-acetylcysteine is having that cysteine part which will be replenishing the glutathione stores. So glutathione amount will be increased inside the cell, which will be further reducing the glycosylation. Actually, these glycation products are formed due to glycosylation. You call it, you will be looking at 3 molecules majorly. So those molecules formation will be reduced and further crosslinks will be reduced. Like a glyoxal, methyl glyoxal, these are the compounds that are responsible for glycation and oxidation. Hmm. Actually, it forms nitrous acid, right? Nitric acid reacts to form. Actually, nitrous acid will be formed, I think. Nitrous acid will be formed, and products. Um, I don't think so. I would refer. But nitrous oxide is common.